Hello, everyone. I'm Christy Riz, Junior Editor at GenomeWeb, and I'll be your moderator for today. The title of today's webinar is The Impact and Regulation of the Ribosomal RNA in Arabidopsis Meiosis, and our sponsor is Lexogen. Our panelists for today are Dr. Jason Sims, a researcher in the Department of Chromosome Biology at the Max Perutz Labs at the University of Vienna, and Dr. Ivan Gopal, Product Manager at Lexogen. You can type in a question at any time during the webinar. You can do this through the Q&A panel, which appears on the right side of the webinar screen. And if you look to the tray at the bottom of your window, you'll see a series of widgets to enhance your webinar experience. So with that, I'll turn it over to Dr. Sims. Please go ahead.
Thanks, Christy, for the introduction. And I wanted to start by thanking Lexogen for inviting me to give this webinar here today. So the story I want to tell you about is about two of my favorite things, which is ribosomal RNA and meiosis. So I'm going to give you a brief introduction into meiosis and what ribosomal RNA is, and then I'm going to dive deep into the data. So before I begin, I think we have to answer an important question, which is why do we study meiosis in plants? Well, meiosis is a very important process that generates genetic diversity. And this is extremely important for breeders that can select for certain traits in their crops. So these are some of the crops I'm sure you came across during your life. We have maize, wheat, barley, and also the infamous broccoli. But we study meiosis in Arabidopsis thaliana, which is a model plant. We use it as a model because it's easy to handle and to work with. And some of the things that we can find in Arabidopsis, we can translate them then to more complex genomes and more complex crops. So this is why we use Arabidopsis to work on meiosis in plants. Now, meiosis is that essential process that after one round of DNA replication, you have two subsequent cell divisions that lead to the formation of four haploid gametes, which you can see at the bottom right of your screen in telophase 2. These gametes are, in plants, will be the pollen and the ovules, whereas in us humans, it will be the sperm and the oocytes. So this is a very widely conserved and ancient mechanism to generate genetic diversity. So I will not go deep into the details of how meiosis work, but I will just pop up now some names of proteins that we use to lead us through the meiotic process. And most importantly, we work on the first stages of meiosis, which is meiotic prophase, which is defined by leptotene, zygotene, pachytene, and biploidine stage. So the first four nuclei you can see at the top. Now, in order for us to identify in which stage each nucleus is, we use certain markers. For example, during leptotene, which is where the double-strand breaks are induced within the DNA. So the DNA is purposely broken by the cell itself, and this is essential in order to generate this genetic diversity. During this stage, we use two markers such as ASY1, which forms foci-like structures, and it's a protein essential for building the meiotic axis, which is needed to basically help the chromosomes to find their homologs and form the so-called synaptonemal complex. We also look at proteins like ROD51. ROD51 is a marker of where double-strand breaks have occurred and have been processed and are ready to start the repair. As we move from leptotene into zygotene, here ASY1 from a foci-like pattern, it will give us more a ribbon-like structure, meaning that the axis has been fully formed along the homologous chromosomes, and we will start to see the appearance of the protein called ZIP1, which indicates a fully formed synaptonemal complex. Here, the ROD51 foci will start to decrease, meaning indicating that the breaks that were generated in leptotene are starting to be repaired. Finally, in pachytene, this is where all or most of the DNA breaks have been completely repaired. ASY1 will almost completely disappear except for a specific region, which I will tell you in a minute. And ZIP1 will almost completely substitute ASY1 and form this ribbon-like structure. And here in pachytene is where the breaks are basically channeled into the forming a crossover. Crossover means you have the physical connection between two homologous chromosomes, and this is essential to generate this genetic diversity during meiosis. After this, you quickly move to diplotin, diakinesis, and metaphase 1, where there's the first division and the homologues separate, and then we have a mitosis-like separation where we separate the sisters in metaphase 2. Now, let's say we have a DNA break in our genome, in our DNA. There's different ways of repairing a break. You can repair it in a very quick manner by using a non-homologous end-joining, or NETJ, or microhomology-mediated end-joining, MMEJ. And these are quick ways to repair the DNA, but they are error-prone. Another way to repair it is if you go through the homologous recombination pathway. This brings to the synthesis-dependent strand naming, or homologous recombination itself. Now, during meiosis, we want to make sure that the error-prone pathway is blocked, and this is mediated by the fact that the breaks are produced by the conserved topoisomerase-like protein called SPO11. SPO11, once it makes the break, it will sit on it, and it needs to be removed from the break site itself, and this will be mediated by MR11, the MRN complex, and COM1. Once SPO11 is removed, the ends can be resected, and we can go into the homologous recombination pathway, which ensures also the formation of crossovers. Now, why is this important? Well, if we look at a plant chromosome, in this case, the chromosome of barley, you can see we have two different maps here. We have a physical and a genetic map. The physical map indicates the actual distance between different genes and how big this chromosome actually is. If we look at the genetic map, it indicates how far these genes are between them genetically, meaning with which frequency they can actually segregate. Now, you can see that the chromosome is, or in the red part, is extremely overrepresented in the genetic map compared to the physical map. This indicates that in the physical map, there are regions which are suppressed for homologous recombination and crossover. And this for breeders is not a very good thing, because if within these regions we have, for example, the genes related to good taste and the genes related to a good color of the crop they're working with, you understand that it will be difficult for them to actually select and recombine these genes if their genetic distance is very, very short. This is why we need to try and understand how we can exploit meiosis to make sure that the breeders can actually recombine these genes. Furthermore, these highly suppressed regions are usually very, very high in repetitive DNA. Now, and obviously this is actually very important also for the current situation we live in, which is global warming, where we want to select for plants that can resist higher temperatures. Now, one of my favorite repetitive regions is the rDNA, or ribosomal DNA. It's this essential region that encodes for the ribosomal RNA, which is the scaffold upon which the ribosomes can be formed. Now, in Arabidopsis thaliana, we have approximately 800 copies of these rDNA genes located on chromosome 2 and chromosome 4. At the bottom, you can see of the scheme of Arabidopsis, we have one rDNA unit, meaning one copy, and this is how it looks like. It has the 18S, 5.8S, and 25S coding genes, which actually go and form the small subunit of the ribosome, which is for the 18S, and the 25S and the 5.8S go and form the large subunit of the ribosomes. And then we have what were known as externally transcribed spacers and non-transcribed regions, which are very important in order to regulate actually the transcription of the rDNA itself. Now here in that red box you see represents a variable region, which we call the VAR region, which is specific and unique to Arabidopsis because it allows us to differentiate which rDNA units are located on chromosome 2 and which are located on chromosome 4. For example, on chromosome 2, we will have the rDNA units of variant 1 and 3A, whereas on chromosome 4, we will have variant 2, 3BC, and variant 4. These RNA units are clustered together in a very large region that's known as the NOR, or nucleus organizing region. Now, these highly conserved repetitive regions are present in many other organisms. We have them in humans, located in 5 chromosomes. You can see that the single unit itself is very similar to that of Arabidopsis. It's just it's just longer. Mice have it as well. We have 6 chromosomes, and also yeast has it, in this case on one chromosome only. In fact, it is believed that the rDNA unit, or the rDNA sequence, is probably the most ancient gene present. But how are these regions actually regulated? Let's say, in a fully developed plant, what is known is that you don't really need, or this is also the case for us humans in general, you don't really need all these units to be transcribed at the same time, because the amount of ribosomes you need for a specific time or within a specific cell varies a lot depending on the tissue that you're looking at. In a fully developed plant, let's say, what you will see is that NOR2, so the rDNA located on chromosome 2, is usually inactive, so it doesn't participate in the transcription and in the pool of ribosomes present within the cell, whereas NOR4 is actually the active rDNA region, so the rDNA present on chromosome 4. And these are the ones that participate in forming the ribosomes. Now, this is not a black and white situation. This changes also depending on the different tissues. We obviously investigated how would this work in a meiotic environment. Now, in meiosis, the first thing we had to figure out was, are the RNA units transcribed? How many in which ones? Now, if you look at on the left here, we have the leptotene panel. The DAPI indicates the nucleus itself, so the DNA. We fished for the 45S rDNA by using a specific probe, and we detected DNA-RNA hybrids, which are basically the byproduct of high transcription, by using the S9.6 antibody. As you can see, that the S9.6 antibody detects or marks all 45S regions within the DAPI stained nucleus, indicating that probably all of the rDNA, or most of it, is actually actively transcribed during meiosis. In contrast, if you look at the somatic nucleus or mitotic nucleus, you can see that only 2 NORs, which are indicated by the white arrows, are the ones that were actually transcribed, because they have a high S9.6 stain. The other 2 have a very mild or almost absent staining, which is reassuring because it shows us that what we know is correct, that only some of the RNA is transcribed in somatic tissue. We didn't only look at this by cytology, we also did in RT-PCR to look at if the variants were actually transcribed. And you can see here the NOR2, which is the inactive one, bears variant 3. And you can see in the bottom gel picture, in early meiotic, so EM, late meiotic, and within fertilized embryos, variant 1 is always strongly present, whereas in the adult leaves, or AL, there's very few or almost no variant 1 present, indicating that NOR2 is probably suppressed within the leaves, nuclei, but strongly present and transcribed within meiotic and within the fertilized calyx, so within the embryos or the fruits of Arabidopsis itself. Cytologically, what we also found was that in pre-meiotic cells, the 45S rDNA is actually located around the rim of the nucleolus itself, which is where the transcription of the rDNA actually takes place. But in leptotene, what we saw is that it actually translocates within the inner part of the nucleolus, which strongly reinforces our idea that all of the RNA, or most of it, is actually being transcribed. Now, this caught our attention because we wondered whether if this region would actually acquire certain features necessary for it to undergo a normal meiotic process. And we looked at, for example, if the RDNA would acquire ASY1, which I told you before, it's a protein necessary for forming the axis and to allow proper crossover formation. And as you can see that the RDNA being recruited within the nucleus itself, the ASY1 doesn't colocalize with the 45S region, indicating that it seems that the RDNA is actually protected from the formation of a proper synaptonymal complex. And therefore, it cannot have crossovers forming. We looked at also the other stages. That was leptotene. Here I'm showing you zygotene and pachytene. You can see also in zygotene, the 45S or the RDNA region will actually fuse and form a unified structure, which is also absent of AS1. We know we are in zygotene because we can see some ZIP1 staining indicating that synaptonymal complex is starting to form. But what was quite peculiar is that the moment we enter pachytene and we have a full pachytene, meaning all chromosomes have completely paired, and this synaptonymal complex is formed all along these chromosomes, only and exclusively during this time point, we see ASY1 colocalizing perfectly with our RDNA regions. And this really caught our attention because it means that this region is actually not regulated like the rest of the genome during meiosis. So we went a bit more into detail and looked at if the base of the axis would actually be there. And this is where we looked at the meiotic cohesion called REC8, which is the base of the axis upon which ASY1 will then build the proper axis, and you will have the synaptonymal complex build. There's here on the left side, I put a scheme of the cohesion, which looks like a ring, and it's necessary. It has many, many functions, one of which in meiosis, it keeps the sister chromatids together, but it also has functions in chromosome organization. You can see the blue half moon line indicates different alpha glycines. In Arabidopsis, we have 4 different kinds. REC8 is the meiotic specific one, and then I will mention one of these 2. But what's important here is that if we focus on pachytene, you can see that highlighted by the white square, you will see that on these ASY1 late stretches that we see, there's very few or almost no REC8 colocalizing. These are images taken at super resolution using STED microscopy. We have around 40 to 60 nanometers resolution here. But if we look at ASY1 and SEC3, which is another component of the cohesion ring, we can see in the bottom square, in the bottom white square, we can see that ASY1 nicely colocalizes with SEC3, indicating that even though there is no REC8 cohesion, meaning no meiotic specific cohesion, we do have another cohesion type region. So I will not go too much into detail here, but what we did, we looked at if actually REC8 is not present. So we quantified if REC8 would be there by using ARIScan microscopy. And you can see at the bottom of the images, we have the line profile that indicates how much ASY1 and REC8 we have present. You can see that REC8 is almost completely absent at the peak of ASY1, whereas SEC3 is much more strongly present, indicating that REC8 is not there during meiosis at these specific regions. But when we collaborated together with the lab of Schnitger, and this is images from Viola Kuting, they approached us and told us that they actually found that specific the specific cohesion that would be recruited to the RDNA, and this is RAD21.2. And you can see the nice pictures from Viola here that really show how RAD21.2 specifically colocalizes with the RDNA, and it's there when there's no REC8. Now, unfortunately, I don't have too much time to go into detail in what happens if we remove RAD21.2 and all the different things that happen to the RDNA if we perturb its stability. But what I would just want to mention is that if you perturb any little aspect of the stability of the RDNA, either it being the chromatin environment or the nucleolus or the cohesion that is around the RDNA itself, this leads to catastrophic events, which means that you would have crossover and recombination occurring between non-homologous chromosomes, because it occurs between the repetitive DNA of chromosome 2 and chromosome 4, so between NOR2 and NOR4. And this leads to these connections that you can see on the right, highlighted by the black arrow. These are not good for the plant itself because they lead to missegregation and also an unbalanced number of copies of RDNA copies within the gametes. So this is one of the reasons why these regions are suppressed for meiotic recombination. Now, here I just want to show you an example of this missegregation. You can see at the bottom left of your screen, you have the 5 chromosomes of Arabidopsis, which we can easily mark and identify by using specific probes that highlight the different chromosomes. We have 5 chromosomes in Arabidopsis. Here, what you see is a telophase 2, so a very late stage where you would then have the formation of the 4 haploid gametes. And here we have the 4 haploid gametes, and if we count the chromosomes within these gametes, we would have 5 and 5 for the top left 2 nuclei. But if we look at the top bottom 2, you will see that we have 4 on one side, and on the far right side, we have 6. And this shows that there has been a missegregation of the chromosomes, and actually the chromosome that has missegregated is chromosome 4, or the NOR-bearing chromosome. So this indicates that if you perturb the stability of these repetitive regions, it generates a high level of genomic instability. So just to summarize this first part, before I go into the sequencing that we actually did, we found that both 45S RDNA loci are transcribed at the onset of meiosis, that meiotic RDNA is embedded in a unique chromatin environment, and that the nucleus shields the RDNA, I didn't have time to show you, also from spoiled-mediated double-strand breaks. The repair of the RDNA depends on NHEJ. Unfortunately, I didn't have time to show you this for the sake of time, but I'm happily discussing that later if you want to. And the RDNA can create an HR-refractory chromatin environment. This is also something that I didn't have time to show you, but I'm happily talking to you about this later if you want during the Q&A session. Now, but we wanted more from this. We wanted to actually go down and see at the sequence level, how does the RDNA look? And it was quite unfortunate that if you go and look at your genome browser of any organism, the RDNA is not there because this region is very complex, it's very difficult to sequence and assemble due to the fact that it's very long and that each unit is also quite large. So it's very hard to get an assembly of this region and to have an idea of how the structure of these RDNAs, how they are organized within this region so this is quite hard to do, let's say. And just recently, a couple of weeks ago, it came out that in humans, they were able to sequence these regions. And we, on the other hand, we sequenced and assembled NOR2, so the RDNA region of chromosome 2. How did we do this? Well, we used a clone-based approach, so a BAC-based approach, which stands for bacterial artificial chromosomes. How did that work? What we did is we used BACs which contain only a certain amount of RDNA units. So it has a fixed amount of RDNA units, not the complete, let's say 400 per chromosome, but just a small amount, which is a controllable way for us to sequence them. We sequenced these BACs both with nanopore sequencing, which gave us the opportunity to assemble them and have the overall structure of each BAC. And we also sequenced them by Illumina sequencing, which gives us sequencing depth. So it enabled us to identify and precisely locate each single nucleotide polymorphism and indel located within each RDNA unit. We sequenced quite a few of these BACs, actually 60 or 59 of them, revealing 405 unique RDNA units. Here, what you see is that by this graph, it shows you the distribution of RDNA repeats, so a number of units we had per each BAC. And I just wanted to they're very, very, very similar. So once we had all these BACs, we needed to find a way to put them together to build nor, like if you would build a big puzzle or if you would build something with Legos. So how to do this? Well, we actually used this variation that we have within each unit, this 1% to 3% variation, to build what we call as a barcode. Now, what we did is by looking at the different features each rDNA has. For example, if you look at the top rDNA unit, you can see that it can have a different amount of SP and CP, which stands for spacer promoters and core promoters, from 1 to 5. It can have the presence or not of a 1 site, which is a restriction site. It can have the presence or not of a 3-nucleotide insertion, which is the cat insertion, or it can be of variant 1 or 3 type. Now, if you look at the variant just under this one, you see that, for example, this one has 2 promoters, so we give it a number 2. It has no of a 1 site. It has no cat site, so it has 0, 0, and it's variant 1, so number 1. So in total, this unit will be defined as a unit 2001. Now, by using this barcode system, then we were actually able to put the box together like a big Lego and assemble, not completely, but at least gives us an idea, a draft assembly of how this rDNA region looks like. We went up to 230 kb long regions, actually a bit more. So we could put, let's say, a line together in a long stretch without gaps around 23 to 26 of these units continuously. This gives us the opportunity then to look at each unit individually and also to look at the different SNPs and indels formed, located within these units, and see how they're organized. Now, I don't have time to show you how we looked at the overall structure, but what we actually did was that we could identify specific variations that were clustered together, meaning units containing similar features tend to be close together. And this gave us the opportunity to build specific fish probes that could identify these clustered units. In this specific case here, what you see, this is a wild-type plant. SAD1 repeat stands for, it marks all of the rDNA repeats. Here, cat plus stands for the fact that we're marking just the cat specific cluster, and you can see that this little green dot indicates that these are approximately 47 units located on chromosome 2. And in DAPI, you see the 5 chromosomes of Arabidopsis. We also developed a specific, on the far right, and a specific qPCR analysis that would enable us to count how many copies we have of these specific cat-bearing units. And if you look at mutants where the rDNA environment is actually perturbed, what we found was that we can actually now visualize crossovers occurring between these repetitive DNA elements. For example, in the RTL1 part of the panel, you can see that both rDNA regions contain these 2 green dots, indicating that there's been a crossover or a migration of information from one NOR to the other. The HDA6 also has a much more complex situation and also an expansion of these cat-bearing units. And the NUC2 mutant also shows an expansion of these cat-bearing units. So this all indicates that there is increased crossovers occurring within these mutant lines. But now, with all of the sequence information that we had, we didn't only use it to build fish probes, but we also used it to look at the individual SNPs and indels located within each rDNA unit. And here, what you see from this graph, NOR4 is the blue circle, the blue ball, and NOR2 is the red ball. And this indicates the frequency with which certain SNPs indels located within one rDNA unit. You can see at the X axis, we have one rDNA unit. It also shows where the 18S, 5.8, and 25S are located. And I'm sure you can appreciate that most of the variations actually occur outside of the coding regions. This is because these regions are obviously under very strong environmental selection, and also because transcription it's known that usually you have a lot of SNPs indels occurring at the transcriptional start sites and transcriptional termination sites of these regions. So we have quite a lot of variation. You can see on the right side, we have a zoom in of the 5.8S region where we have the AVA1 in the cut side. And here you can see that the red ball of NOR2 is much higher in frequency than NOR4, indicating that the cut-bearing rDNA units are predominantly present on NOR2. But we also zoomed in the 18S and 25S rDNA regions. And here you can see that the frequency, even though it's very low, it's still present. So we still have, for example, in the 18S, we have around 2.5% and lower amount of frequency of SNPs indels occurring at a specific position. In the 25S, we can go up to almost 4%. These are very low occurring SNPs indels, but still they are present in the DNA. And this is where we sort of questioned whether these would actually be present in the RNA itself. And here we use the Coral kit initially to do total RNA sequencing, and this is the complete picture that we get. Here on the left side, you have the 18S and the 25S marked in green and in magenta. The numbers right next to it are the coordinates of where these SNPs indels are located. And then we have WG, which stands for whole genome. So this is the SNPs indels located within the DNA. And then next to it, we have different tissues. So NOR2 shows you the SNPs indels located only exclusively on NOR2 within the DNA. And then we have AL, which stands for adult leaves, YL, young leaves, inflorescences, and calyx. And this shows you the pattern of how these SNPs and indels are actually transcribed. So we only focused on those SNPs indels that we found in the RDNA, in the RNA, excuse me, which are also then found in the DNA itself. This indicates that if you have something in the DNA, you should find it then in the RDNA, in the RNA if it's transcribed. And you can see here that there is a slightly different pattern and also in the transcription, and this is tissue specifically changed. So you can see that in the adult leaves, for example, the bottom part of the graph shows you that these variants are almost completely not present in the adult leaves, but you can strongly see them, for example, in the young leaves. In the inflorescences, for example, we only have one specific SNP indels appearing. But this is total RNA. We're looking at also non-processed RNA. We wanted to go even deeper, and we wanted to focus specifically on the ribosomes themselves. Do ribosomes have ribosomal variants which are tissue specifically expressed? So what we did here, always with the Coral kit, we specifically enriched for ribosomes, and we did this by using UV cross-linking the ribosome, so we locked it into place. Then we used a magnetic bead bound to specific probes that would bind exclusively to our ribosomes, and then with a magnet we enriched for our ribosomes from our plant slurry. Just to show you that this protocol works quite well, we have an almost 8-fold increase in ribosomal RNA if we use our system after cross-linking. If we use a control magnet bead, which has LNA probes that do not bind to the ribosome itself, or if we don't cross-link the ribosome, we get almost no ribosomal RNA out. And the bottom graph in purple and in cyan, you can see that in the total RNA, we also get a lot of reads that map to these non-spliced out regions of the RNA, so what's called the pre-processed RNA. Whereas with our ribocapture system, we get almost no or very, very few reads that bind to this region, and most of them, or the majority of them, bind to the actual coding sequence of the RNA itself, indicating that we're pulling down on full intact ribosome, or at least on the subunits, the process subunits of the ribosomes themselves. And what are the results? Well, what we found out is that here again, each number indicates the position where the SNPs indels can be found. The first line you see, whole genome, indicates the variation located in the DNA, and then you have only RNA sequencing, adult leaves AL, young leaves YL, inflorescences, and leaks. And you can for sure appreciate that, firstly, the number, all the SNPs located within the DNA itself, only very few of them are actually found within the ribosomes, indicating there's a very strong transcriptional control of which rRNA variations will be integrated into the ribosomes themselves. Furthermore, what I'm sure you can appreciate that there's some variants which are always present in all tissues. I'm talking about the first two lines at 5736 and 668 position, 6680, sorry, position. And if you look at the bottom part, what you can see is that there are certain variants which are actually tissue-specifically expressed. For example, 7440, you can only see it present in adult leaves and within inflorescences. So this indicates that there's a very strong transcriptional control and that we have tissue-specifically present rRNA ribosomal variants. But now, I'm sure some of you are questioning, but what about meiosis? What is this all about with meiosis? Well, we went with the next step, and this is where we collaborated directly with Lexogen. We used a Thor chemistry combined with the Carl, because this gives us the opportunity to not be 3' biased in our sequencing experiments, and we could recover 30% of the reads. And we did this at a almost single cell level. So here what you see is this panel indicates the variations in this case contained in the 18S rRNA. On the left, as always, the numbers indicate the positions of where these variations occur. You can see the variations occurring in the DNA. And then we have three controls: adult leaves total RNA, adult leaves pull down, and adult leaves single cell sequencing. So you can see we have certain variants that appear in these three controls. Then we went a step further, and we went to sequencing, I would say not completely single cells, but we had between 3 to 5 single meiotic cells. G2 is the cells just before they enter meiosis. It's another control. And then you have lepto, which stands for leptotene. Leptocolumn is a mix of around 50 cells. And then lepto 1, 2, 3, and 4, these indicate 4 replicates of 3 to 5 individual meiotic cells. And here you can see that we have the appearance of a specific variant, for example, occurring at the position 3321. As we move from leptotene into zygotene, you can see that there seems to be a switch actually in the variant that's present at this specific stage. So we see more predominantly present 3238, sorry. And then as we move from zygotene into pachytene, you can see that variants are completely disappear. And then here you have 3 controls, which are a mix of, I would say, thousands of meiotic cells, which were then sequenced by total RNA sequencing. So not using just the Thor plus Coral chemistry. This is the 18S. If we look at the 25S, we have a very similar picture here again. And you can see that in this specific case, we have the presence of meiotic-specific variants, which are always present in leptotene, zygotene, and also pachytene. So here in this case, for the 25S, the appearance of these meiotic-specific variants remains present throughout the meiotic process. They're fortunately not found in the control, but this was a different experiment. Now, in the second summary, and before I bring you to the outlook of what we actually did further, we sequenced 405 individual RNA units, which were assembled into large contigs. These RNA units are organized in clusters with similar features. Individual clusters can be visualized by FISH because we know the sequence and we can find them now. The ribosomal variants are expressed in a tissue-specific manner, which means that we have a heterogeneous population of ribosomes at the rRNA level, and only a small fraction of these ribosomal variants are transcribed at the given time, which means transcriptional control. And we also have meiotic-specific variants. Now, for the last minutes that remain for the talk, I just want to show an outlook. In collaboration with Lexogen, we didn't only use the Thor plus Carl's kit for sequencing single meiotic cells, we also used the Luther-based system. In the Luther-based system, you're 3' biased, and you also, in this case, you have very low number of rRNA reads, so around 0.8%. You can see that here in this table, the raw number of transcripts and genes is pretty much actually identical between the two systems. But if you focused on the expressed transcripts, which is at the bottom of the two tables here, you can see that in the Thor plus Carl system, we have around 40,000 expressed transcripts that we can detect, whereas in the Luther, we can detect around 28,000. This is because the Thor plus Carl system enables us to not be 3' biased and get the full length mRNA transcripts, and we can detect different expressed mRNA transcripts during meiosis in different meiotic stages. We looked at in detail actually which of these genes would be expressed. We did here this in this volcano plot. What you see here in red is a contrast groups between leptotene, zyglutine in contrast with pachytene. In green is leptotene and zyglutine, and in cyan it's zyglutine and pachytene construct groups. You can see we have quite a large number of genes which are differentially transcribed, but we also have a large number of differentially transcribed transcripts. And what we actually did is step further. We actually looked at some of these transcripts, and we didn't go too much into detail which genes they are. I just mentioned that there are genes related to crossover formation that we found, and we also found genes related to the ribosomes themselves. You can see that the left panel shows you we found 53 differentially expressed genes, and on the right panel we found 56 differentially expressed transcripts, which means that we had splice variants that were differentially transcribed throughout meiosis. At the bottom, we have leptotene, zyglutine, and pachytene, and I'm sure you can easily appreciate the differences specifically between pachytene and also the other stages. Also, zyglutine has a very large number of genes which are differentially transcribed. So this indicates, in general, that there's a very tight and a very strong regulation and transcriptional difference throughout also the different meiotic stages, so leptotene, zyglutine, and pachytene. Now here, I will end my talk by thanking all the people that worked with me throughout for this project, actually. So I work in the Ribosomal lab. Here on the right side, you can see a small GIF animation of us in the Max Perutz terrace, nicely social distancing with our personalized masks. I have to thank, obviously, the current members of the group, which had created a great work environment, specifically Peter, which is my mentor for these years. Past members, Giovanni Sistini, which worked with me on the sequencing of the rDNA. Collaboration partners, Art von Hisler from the Max Perutz, that gave us the sequencing, the bioinformatic power for us to analyze our data, and also our Schnitger and Viola, which we collaborated with them actually in the RAD 21.2 story, which was great to work with them. And finally, also Yvonne from Lexogen, that she was my direct contact for Lexogen and for all the sequencing stuff. And I have to thank my funding. And with that, I end my talk, and I'll be very happy to take any questions if you have them. Thank you very much.
Thank you, Dr. Sims. As a reminder to webinar participants, if you have a question, please type it into the Q&A box in the control panel. We'll conduct the Q&A portion of the webinar after the presentations have concluded. Next up is Dr. Gopal. Please go ahead.
Thank you, Christy, for the kind introduction, and thank you, Jason, for the great talk. I will take a few minutes to introduce the Lexogen technologies that Jason used in his project. The first up is Coral. Coral is a whole transcriptome library prep for transcript level analysis. It uses a fragmentation-free protocol with excellent strandedness and exceptional representation of transcriptional start sites, as you can see here from the gene body coverage plot obtained from an endogenous transcript. Coral contains unique molecular identifiers for the removal of PCR duplicates and for highly accurate quantification. The UMIs are introduced in the second reaction step without the need of additional reagents, and therefore the streamlined workflow can generate RNA sequencing libraries in just 4.5 hours. Coral is also suitable for low input samples, for degraded RNA, and for FFPE samples. The gene detection plot here shows excellent gene discovery rates across the wide range of RNA input amounts. Here, 1 to 100 nanograms were used prior to rRNA depletion. On the right side, you can see gene discovery rates comparing FFPE and fresh frozen samples, which are also highly comparable, and qualitative analysis shows a roughly 90% overlap of detected genes between the two sample types. Coral is also available with UDIs, so with unique dual indexing for maximization of the sequencing output. The second technology that was used is Thor amplification, which is actually a part of the Luther 3' mRNA-Seq library prep for single cells. But before we come to Thor amplification, I would like to quickly introduce Luther. Luther is a 3' mRNA sequencing kit to study gene expression in single cells and in low-input RNA samples. It can also be used for challenging samples. Luther uses a novel direct RNA amplification technology, which is termed Thor, and this yields unprecedented sensitivity and reproducibility for single cells. So how does the Thor amplification work? A T7 promoter is introduced by using a primer with an oligo-DT stretch to the 3' end of the endogenous mRNA molecule. In a series of reaction steps, the protruding 3' tail from the endogenous RNA is removed, and the T7 promoter sequence is fused to the original RNA molecule. By this, the T7 promoter is also converted into a double-stranded active promoter. And Thor amplification then uses T7 polymerase to initiate the transcription of the endogenous original RNA molecule. In vitro transcription generates antisense RNA copies, which are shown here in light blue. And the original mRNA is thereby repeatedly copied over and over again to create amplified RNA. The major novelty here is that the original RNA molecule is used and not a cDNA intermediate that is used for conventional single sequencing. The amplified RNA can then be converted into 3' mRNA-Seq libraries by using the Luther kit. As shown in Jason's talk, the Thor amplified RNA can also be used as input to whole transcriptome library preps such as Coral, and this allows also alternative splicing analysis. With this, I would like to end my short talk and thank you all for listening. And we have a short announcement before I hand over. We have recently launched our RNA Expertise Hub. That is an e-learning resource. The hub contains short videos on technology, also including Luther and Thor amplification. It contains a download link and our RNA lexicon. You can find out more by scanning the QR code or following the link below. And with this, I would like to hand back to Christy and Jason for the Q&A session.
Great. Thank you, Dr. Gopal. As a reminder to webinar participants, if you have a question, please type it into the Q&A box in the control panel. And before we begin the Q&A, we'd like to ask attendees to take a moment after the webinar has ended to give us feedback by taking our exit survey. All right, so our first question, Jason, since homologous recombination is suppressed at the rDNA, what repair mechanism takes its place in this region?
Yeah, so I didn't have time to show you this, but what we found is that NHEJ, so non-homologous joining, is the one that actually takes over the breaks within the rDNA. But I must say that predominantly the breaks that are made within the rDNA itself during meiosis are not SPO11 mediated, which means that NHEJ can easily repair these breaks because it doesn't have SPO11 that impedes them at the ligation part, I would say.
And what do you think is regulating the expression of the ribosomal variants?
This is a very good question. This is actually for the future, so I don't have a direct answer for this. But I didn't have time to show you this, but I showed you that we have in the RNA units, we have different amounts of promoters, spacer and core promoters. Now, these spacer and core promoters are known to be actually active and generate what are known as small RNAs, which mediate the RNA-mediated methylation of certain regions. And this would actually silence predominantly or not certain regions. So I think it might depend on how these promoters are activated or during meiosis, but we still have to work on this. This is just speculation.
A point of clarification on the slide where you showed SNV percentages, was that cell variation, or was that some cells carry those SNVs and others don't?
No, that's you mean if you mean in the part where I showed the RNA unit with all the different variants occurring within each unit, this is the global amount of variations which occur within the genome and within the entire organism.
Okay, great, thank you. So why did you decide to use the Thor chemistry to detect the ribosomal variants?
So we use the Thor and the coral combined because let's say the bottleneck of this setup is that we have very little material, which is just a couple cells, and the Thor plus the coral combined gives us the opportunity to really go down and sequence 3 to 5 cells only, meiotic cells only. So this is why we decided to go for this.
All right. I believe that those are all the questions that we have in our queue. So we're going to wrap it up there. Thank you, Jason Sims, Yvonne Gopal, and our sponsor, Lexogen. As a reminder, please look out for the pop-up survey after you log out to provide your feedback. If you missed any part of this webinar or wish to listen to it again, a link to an archived version will be emailed to all attendees. Thank you for joining us for this Genome webinar.
Thanks, everybody.